genetics. And this directly relates to, obviously, reproduction, because we've already talked about the formation of the egg and the sperm and how the chromosomes behave during that formation. And then we also talked about a little bit about fertilization and the possibility of producing twins, like monozygotic versus dizygotic twins. So we're going to go into a little bit more detail about how traits are passed from one generation to the next. And when we look at the human genetics, there was recently, in the 1990s, there was what was called the Human Genome Project. So you might have remembered that. Okay. And the Human Genome Project, the idea was to sequence the DNA. So DNA is composed of nucleotides. So the purpose was to sequence the DNA. And DNA is a type of molecule that's called a nucleic acid. And it's composed of nucleotides that are abbreviated A, T, C, and G, right? So when we talk about the sequence, what we're doing is what we're talking about is the sequence of letters, right? So when we look at this sequence, there's in the human genome, there's 3 billion what are called base pairs. Okay. And so what the base pairs refer to is the fact that the DNA is double-stranded, and that we have pairing of nucleotides, so that if we look at we looked at a sequence like this, right? This would be a sequence of DNA. The complementary sequence, A pairs with T, T pairs with A, G pairs with C, so it might look like this. And so this would be a sequence of DNA that is double-stranded. So I just wanted to point out that there's actually there would actually be 6 billion bases or 3 billion base pairs. Okay. So this was actually a really interesting historical project, because at first it was the Department of Energy that took on the sequencing of the human genome. And when you think about the Department of Energy, like, why would the Department of Energy, DOE, be big into the Human Genome Project? Does anybody know, or maybe hypothesize why the Department of Energy would care? So one source of energy is nuclear, right? And so nuclear energy sometimes releases radioactivity, and radioactivity can cause mutations in our genetic material. So they were interested, the Department of Energy was interested in the Human Genome Project so that we could better understand how mutations affect the outcome of what the genes produce and how they interact with one another. So it was the Department of Energy. And then National Institutes of Health, or NIH, became involved in it. And then it was kind of interesting because it was deemed that the government was being too slow. So it was going to take them forever to finish the Human Genome Project, and this is in the 1990s. And so there were some startup biotechnology, like for-profit companies, that became involved in the Human Genome Project. And it was of concern because the idea was that these for-profit companies were going to sequence the human genome, and then they were going to patent their information. And what that means is that in order to use the information, then you'd have to pay these biotechnology companies. And so it was a huge deal, and finally the government started working with the for-profit companies and agreed that they would help them and they would kind of collaborate, but there wouldn't be any patenting of human genes. So it's kind of been overthrown. And the first one was the one that causes the really bad breast cancer, the BRCA1. And so the idea was they found it, and so if you wanted to do research on that gene, you'd actually have to pay them money to do research. And so they found that was kind of unconstitutional to put a patent on a normal gene that is found in the human genome. Okay, So when we look at the Human Genome Project, one of the most amazing things that came out of it is the fact that there are far fewer genes in our genome than we thought. And so if we look at what a gene is, let me define what a gene is first. A gene is a piece of DNA that codes for a protein. So that sequence of letters can be translated into a sequence of amino acids, because proteins are obviously not made of nucleotides or bases. They're actually made of amino acids. Okay, so a piece of DNA that codes for a protein, and we have, you know, over 100,000 proteins in our body. And so the idea is that if a gene codes for a protein, there should be as many genes as there are proteins. So one of the most amazing things that came out of the Human Genome Project is that we actually only have about 21,000 genes. And so we're going to talk about this, not today, but on Wednesday, we'll talk about how 21,000 genes could then possibly code for those proteins and what those genes do. Okay, so one gene does not seem to equal one protein in our human genome. Okay. The other really interesting thing about this is because we have so few genes, is only about 2% of the genome seems to be functional. So 2% of our genome codes for proteins. So the biggest mystery now that is trying to be solved is, what is the other 98%? Why is it there, and what could it possibly represent? Okay, some people in the past have called it junk DNA, but it is believed now that there could be important regulatory functions, so that that 98% could regulate the turning on and turning off of the genes in our genome. Okay. Okay. So when we look at how genes are passed from parent to offspring, this is the study of heredity. And so we're going to look at first, what are the sources of genetic variation? So the first source that I want to talk about relates to the formation of gametes. So we'll talk about the formation of gametes. So remember that the gametes are the egg and sperm. And remember that the gametes carry half of the genetic information. So in us, that would be 23 total chromosomes. So our species has 23 total chromosomes in the gametes, and those gametes are said to be haploid. So they have half of the genetic material. So this means that they have 23 total number of chromosomes, so that there are no pairs in the gametes. Okay. So when we look at the meiosis, which is the type of cell division that gives rise to the gametes, we can see that one of the things that leads to an increase in genetic variation is the way that chromosomes exchange genetic material during prophase 1. So we have an event which is called crossing over. Okay. And so this is the exchange of genetic material between replicated homologous chromosomes in prophase 1 of meiosis. So remember that meiosis is the special type of cell division that gives rise to the egg and the sperm. The other type of cell division that is involved in embryonic development is called mitosis. Right? We talked about mitosis in 231 when we were talking about cancer, for example, and cell differentiation. Okay. So that's as prophase 1. Okay, so that's the very beginning of meiosis. And so what this means is that genes that are located on the same chromosome can be then separated. And so in some meiosis, you'll see, okay, so this is the difference between meiosis and meiosis. This is the replicated pairs of chromosomes. And this would be prophase 1. And this is where you have the exchange of genetic material. Okay. So then if we look at a diagram here, and I apologize, for some reason one image got put on the other. I just want to point out that in your book, you'll notice that chromosomes have like weird coloring. So notice how this chromosome has a little piece of purple on it, but it's mainly green. And then this chromosome is mainly purple, but a little bit of green. That is how they represent crossing over, or the exchange of genetic material. So each of those would represent chromosomes that were inherited from your mother and your father in your body. And then during prophase 1, they exchange genetic material. And notice how they just show one arm, but really it would be like taking a red deck of cards and a blue deck of cards and then shuffling them. Okay, so it occurs all over the genome. And then if we look at here, this is another diagram. Okay, so let's say that we had chromosomes that had little h's and little e's, and chromosomes that have big h's and big e's, and we'll talk about what those mean in a minute. But notice here, when we have crossing over, we then increase our variation. So notice how now little h and big E can be inherited together, and big H and little e can be inherited together. And so crossing over is really important for increasing the variation that we see in the gametes, in the egg and the sperm. Okay, so that's one way that we get variation. So the other way that we get variation in meiosis is that we get the lining up of the chromosomes during metaphase 1 and 2. So another source of variation is that the chromosomes line up not independent, well, they are independent of one another, but they line up differently. I'll just see, chromosomes can line up, there we go. Chromosomes can line up differently in meiosis, or excuse me, metaphase 1 and metaphase 2. So metaphase is where the chromosomes line up in the center of the cell. And the way they line up, if you think about it, we have 23 pairs, right, of replicated homologous chromosomes. And so in the diagrams, they only show like 4 pairs, right, or 2 pairs. And so there's not a lot of variation, but if you think about 23 pairs, you could have green and then purple and then green and then purple, green, red, or it's not red, green, green, purple, right? And so they could line up independently of one another. And so that can increase your variation too. So this is what that diagram shows right up here, okay? So this again just shows 3 pairs of chromosomes. And this would be in metaphase 1 of meiosis 1. And I know that because the chromosomes that are separating are not being pulled apart into individual chromosomes. So just to kind of ignore this down here. But if you notice here, they can line up in this pattern, they can line up in this pattern, and this pattern. And then as they proceed through meiosis, then that is going to give you variation in the gametes, okay? So the way that they assort, right? So this is sometimes referred to as independent, so you can write that down under that. This is called independent assortment of chromosomes in meiosis, right? And so that is going to lead to more variation. Oops, I didn't spell that right. Independent. Sorry, independent assortment. Okay, So another source of variation, so if we put 2 here, right? Another source of variation is that we have random fertilization of the egg by the sperm, okay? So this would be, generally, it's random. I'll just put a question mark here because we'll talk about an instance where it might not be exactly random, right? But random fertilization of the egg by the sperm. So which sperm is actually the one that gets to the egg and is able to fertilize it can increase the variation, right? So because there's variation in sperm and then what sperm actually makes it through, that would also be a source of genetic variation in our offspring. Okay, so I'll put as an aside here with this question mark, right, is that sperm with a Y chromosome instead of an X, which we'll talk about what that means, right? Sperm with a Y chromosome are generally faster than sperm with an X chromosome. So those are the sex chromosomes. And that's why they say that males determine the sex of the offspring because they can produce a sperm with a Y chromosome or a sperm with an X chromosome. And so the Y chromosome is smaller. So this the idea here that they're actually a little bit lighter. I don't know if that's true, but it seems that sperm with the Y chromosome fertilize the egg more frequently than one with the X. So what that means is that there's actually more baby boy embryos produced than baby girl embryos. And then we'll talk about why the sex ratio evens out as we see which individuals survive and reproduce. Okay. So those are 2 major, actually more than 2, but the sources of genetic variation. Okay. So when we look at traits, we can say that we have genes, right? And then we have alternative form of genes. So for example, we could have the gene big T, right? So that would be, excuse me, a single gene that codes for a protein. And then the alternative forms of the genes are called alleles. And so in this particular instance, we could have a big T allele, or we could have the little t allele. So when we look at human genetics, there are some traits that we believe to be coded for by a single gene. And one of them is the ability to taste a bitter chemical. And so this bitter chemical is abbreviated as PTC. So this is a bitter chemical. Okay. The ability to taste the bitter chemical is due to a single gene. Okay. So this single gene codes for a receptor on our taste buds that is able to bind the chemical and then cause an action potential to be generated. And then that information goes into our brain as a bitter taste, and we perceive it as being bitter. So we have 2 different alleles for this. So the big T is said to be the dominant allele. And this allows you to taste. And so this would be a taster, right? So you can taste the bitter chemical if you have that allele. The recessive allele is said to be the recessive, excuse me, the little t is said to be the recessive allele. And it is the non-taster. Oops, non-taster. Right. So these genes are located, the T gene is located on a chromosome. So we can put it on a chromosome, right? And everybody in their cells has 2 copies of a chromosome. And so this could be big T, right? Or you could have this combination, big T, little t, or you could have this combination, little t, and that's supposed to be in the same location and on the same size chromosome, so little t, okay? So this is what we call the genotype. So this is the combination of alleles, and I just drew those alleles onto a chromosome because you need to understand that the way that chromosomes behave during meiosis determines how they move into the gametes, and then it determines how the trait is inherited, okay? So if the genotype is big T, then we say that that genotype, actually I write it here, this is called homozygous dominant. So homo means same, zygous, okay? So this one, and there's 2 big T's, so it's dominant. This one is said to be heterozygous because it's a big T, little t. And this one is said to be homozygous recessive. Okay. So that would still be the genotype. So I could say a genotype could be big T, which is homozygous dominant. It could be big T, or little t, okay? Then we can look at the phenotype. Okay, so we could have, oops, excuse me, this individual would be a taster. This individual would be a taster. This individual is a non-taster. Okay, so the reason why we call the big T dominant is if it is present, it is expressed. And so it masks the expression of the recessive allele. Okay. So if we look at the inheritance of this, there's actually a really interesting website called learn.genetics out of the University of Utah, which has got a lot of information about genetics in it. And so they talk about the genetics of the bitter taste. And so the bitter taste actually codes for a protein receptor that is on the taste bud that binds the chemical. And if it can bind the chemical, then it will send a signal to the brain that you can taste it. Okay. And then if we look at kind of the evolutionary explanation for this, is that when we look at toxins that are produced by plants, it is believed that, or it is known that a lot of these toxins have a bitter taste to them. And so plants, when they're bitter, if you think about if you were out in the woods and you were trying to figure out what you could eat and you were hungry, if you tasted something bitter, you would probably spit it out, right? Because you go, oh gosh, that might be poisonous, right? And if it's not bitter, you would probably eat it, right? And so the idea is that bitter taste is, and being able to taste it, is an evolutionary adaptation that would allow you to survive, right? But now it's not that big of a deal because we go to the grocery store, right? And we buy stuff that we know is not going to be toxic to us, okay? So is anybody interested in tasting? Does anybody know, anybody interested in tasting? See if they're a taster or non-taster? Has anybody done it before? Yeah. Yeah, okay. So you just take one little piece of paper, okay? It's like a matchbook. Okay. And I also have cough drops, because if you're a taster, you might want a cough drop afterwards. It's not that bad. If you like dark chocolate or coffee, it'll probably be better. Let's taste it. Let's taste it. Okay. If it tastes just like a piece of paper, you are a non-taster. You're a non-taster. You're a non-taster? Okay. So what is your genotype? You tasted it? No. Okay. Does anybody else want to taste it? I'll try it. Okay. Tiffany's like, no. I'm not going to. Are you a taster? Yeah. Yeah, tasters don't ever taste again. It's not that bad. Okay. So if you are a non-taster, looking at your notes, what is your genotype? Little t, little t. Okay. Are you a taster? Oh, that's interesting.
Wow.
Because you'd actually predict that there would be more tasters than non-tasters in a population because, does anybody want a pop drop?
This reminded me of it kind of irrelevant. Oh, so I don't even know how it came up, but someone was talking about asparagus and how you can smell it.
Yes.
And it's actually based on genes,
Right.
if you can or not.
Yes.
So because I can't, I was like, oh, not even enough. I went around the hospital asking people if they could.
Yes.
Not very many can.
Right. Yep. Okay. So that's just one example of a trait that's thought to be determined by a single gene. And so if we looked at how this would be passed down, so this is actually assuming that that if you were a female and you were heterozygous and you crossed or you had children with a male that was heterozygous, then you could predict what is the variation in your offspring. And so notice how during meiosis, what is going to happen is these chromosomes are going to separate. And so on average, half of the eggs are going to have big T and half of the eggs are going to have little t. And on average, half of the sperm are going to be big T and half are going to be little t. And so this is what is referred to as a Punnett square. And so a Punnett square is just where we combine the gametes to determine what the probability of getting a particular trait would be. Okay, so if we look at this cross, you can write this in your notes. So this would be a cross. This is how you would write it. Okay, so remembering that these genes are on chromosomes, what would happen is that half of the gametes, and I'm just going to draw them like right here, half of them would have big T on average and half would have little t because of independent assortment. And then half would have big T and half would have little t. And then what we would do is we would combine those gametes in what is called a Punnett square. Oops, two N's. Punnett square. Okay, so big T, big T, big T, little t, big T, little t, little t, little t. Okay, so the important thing to note here, so I'm going to draw what all this means so you can remember. Okay, these are the parents. Right, these are the gametes. Right, these in the Punnett square are the possible offspring. So if I gave you a problem, like on the take-home quiz, and I said, okay, so if the parents are this genotype, what is the probability that they would have this? And what would be the probability that you would have this combination, okay? So let's say these both are tasters, right? So this is their genotype, but this individual is a taster and a taster. Right? So what is the probability that they would have an offspring that cannot taste the chemical? 25%, right? So if this is the 4 possibilities, 1 out of 4, right, would not be able to taste it. 75% would be able to taste it, okay? So 1 out of 4. So this is just an easy way to do the probabilities, to multiply the probabilities so that you can figure out what combination is going to be more likely, okay? Okay. So in your book, there's a diagram of some other examples of inheritance of single traits. And some of these are traits, but some of them are also diseases. So if you look at Huntington's disease, albinism, Tay-Sachs, and cystic fibrosis, those would be diseases. Maybe albinism is not more like a trait, but those would be traits or diseases that would have a simple pattern of inheritance similar to the one I described. Now, lots of diseases, like you say heart disease, for example, is not determined by a single gene, right? But it could be multiple genes, and then you can have gene-environment interaction. So Alzheimer's, has genes that predispose you to getting it, but it's also the way that the environment interacts with the genes that determines whether or not you're actually going to get the disease. And we'll talk more about that on Wednesday, okay? So these are just some examples. Okay. So, let's see. We need to talk about modes of inheritance. Right, okay. Modes of inheritance, okay. So we can talk about a trait could be autosomal. And what this means is that it is on a non-sex chromosome. So this would be 1 to 22 pair, right? So the 23 pair is, the 23rd pair is the one that's considered to be the sex chromosome. So autosomal, when you say that a trait is autosomal, that means it's just determined by a gene that is not on a sex chromosome, okay? So this is versus a sex-linked, so this would be versus a sex-linked trait. Okay, so this means that it is caused by a gene that is found on one of the sex chromosomes. Okay, so it could be either Y or it could be X. So we could say that you could have a Y-linked trait, or you can have an X-linked trait. Okay. Now, Y is a much smaller chromosome, and it's only found in males, and so it's generally more rare to have a trait that's on the Y chromosome just because it has fewer genes. Like, I don't remember exactly how many genes that Y chromosome has. Like, I can't remember, 150 or something like that. The X chromosome is big, right? So it has lots of genes. So this is more common, right? That's more common because there's more genes. One thing that is Y-linked is sex. So when we look at the Y chromosome, there is a gene that determines the sex of the offspring. And so we'll talk about that in a minute, okay? So it is more common to have X-linked traits, okay? So when we talk about modes of inheritance, we also talk about whether or not the trait is dominant or it's recessive. So we could talk about dominant versus recessive. So if it is dominant, that means that you only have to have one copy of the gene to have the trait, right? One copy of the dominant allele. If it is recessive, that means that you have to have two copies of that recessive allele. And so when we look at dominant traits, they do not have, we don't see carriers, we do not see individuals that are heterozygous, but when we look at recessive traits, we can see carriers, and I'll explain that in a second, okay? So we can talk about using these terms together. So for an example, we would say cystic fibrosis is autosomal recessive. Okay. We could say Huntington's disease is autosomal, I'll just put a little thing there, dominant. We could say that hemophilia A is X-linked recessive. Okay. So that is important information that I would give you in the question as to what is the pattern of inheritance of that trait, or in this case, of the disease. Okay. So you put those two together, the idea of autosomal and X-linked or Y-linked or sex-linked, and then dominant and recessive. Okay, so in humans, we generally look at patterns of inheritance within families. And in order to describe that pattern of inheritance, we construct what is referred to as pedigrees. And so pedigrees show how traits are passed down through the generations in a family. Oops, wrong way. Okay, so let's look at pedigrees. So pedigrees, if you have a circle, this means that it is a female. If you have a square, this means that it is a male. And then if you color these in, that means that they are the trait of interest. So if I color them in, this means that they have the trait of interest. So that would be phenotype. So this actually just, the pedigrees just show the phenotype or the outward expression of the combination of alleles, okay? So we draw a pedigree in terms of generations. This is a male and the female in the first generation, and they have children, right? And then these children have children of their own, okay? So the way that we designate individuals in this pedigree is we would say that this is generation Roman numeral 1. Right? This is generation 2, and this is generation 3. And then what we do is we number them. And it doesn't matter if they're married into the family, we just number them across. Right? So if I said individual Roman numeral 3-1, you would say, oh, that would have to be this individual right here. Okay, does that make sense to everybody? Okay, so if we look at the ability to taste, right? So let's look at PTC taster. So I want to do a Punnett square of the ability to taste PTC. Okay, this is not sex-linked. It is autosomal, right? So PTC taster. Okay. Now, is the ability to taste dominant or recessive? Right? It's dominant, right? So this is one. So this is autosomal dominant. So when I do a Punnett square, I always create a legend for myself just so that I can remember, right? So what I'm going to do is I'm going to create a legend, and I'm going to say this is the T gene, right? And so I'm going to say big T is taster, and little t is non-taster. Now, which individuals in this pedigree do I know for sure? The tasters or the non-tasters? Which ones do I know for sure what they are? Tasters or non-tasters? What do you do?
Would it be the most recent generation?
No, so is it the ones that are colored in or the ones that are not colored in? Non-tasters, right? So I can go through and I can say, this one has to be little t, little t. This one has to be little t, oops, little t, little t. Oops, because these are the non-tasters, right? And it's a recessive, so we know that they have to be non-tasters, okay? So then, from that information, I can then kind of figure out what I can do. I can figure out what those individuals have to be, right? And so here, right, this one always gets a little t from this parent, but they're a taster, so they have to be heterozygous, right? They could not have two big T's because they have a parent that is a non-taster. Does that make sense to everybody? Okay. And then the same thing with this one. This has a parent that's a non-taster, so you work backwards and forwards, actually, right? And so this one, right, They have a child who's a non-taster, so what must be their genotype? Big T, little t, okay? So I figured out all the genotypes. The genotypes would be the combination of alleles. The phenotype would be able whether they taste or not. So it's the outer expression of that combination. Okay, so let's look at another example. Okay, so this is an example of albinism. So albinism is the lack of pigmentation in the skin, which can be detrimental because that means that they're more prone to damage by ultraviolet radiation. So they're like more prone to skin cancer, for example. And it has a pattern which is said to be autosomal recessive, right? So I'm just going to use the letter A because for albinism, right? And so A, is this going to cause albinism or not? Okay, it's autosomal recessive.
Oh, so no.
No, right? So this would be normal, right? Little a is going to have the disease. Well, we'll just put albinism, right? Albinism. Okay. So the legend is extremely useful here because now which one do I know for sure? People with albinism or people without albinism? With albinism, right? So I know that this person has albinism, so they would have to be little a, little a.
But if they have the A, why is it colored in? They have the big A. Because if they have the trait. Right?
It's recessive. So that means that big A is normal,
Okay.
right? So if I am,
So whoever has the interest has the disease.
so I am interested in albinism, right? I'm in that trait, so I color in the individuals with that trait.
Okay.
Yes. Okay.
All together.
Yeah. So I'm interested in the inheritance of albinism, and I want to know whether or not it's dominant or recessive, and this is recessive pattern, okay? Okay, so if they have two little a's, what would have to be their parents? Yes. And so this is said to be a carrier. So they are carriers. There are no carriers when we're looking at a dominant trait. So these are carriers because they carry the recessive gene, but it is not expressed, right? You cannot tell by looking at them that they have the gene, right, or that allele. They are normal, okay?
What if one parent was the carrier and one parent was not? Then
Well, you wouldn't be able to tell the difference. So people can be like you could be a carrier of albinism and I wouldn't even know it, right? Nobody would know it unless you had a child with another carrier and then all of a sudden you see it come out, right?
So
Okay.
if one of the parents was dominant, or they had the big A, they would definitely, all their offspring would have,
No.
well, 75%.
If one person was big A, so they would be normal, right? They would have no albinism. And you crossed it with an individual that was big A, little a, there would be no possibility of them having an albino child.
So if that was little A.
Okay. Still, you get one gene from one parent and one gene from the other. Okay. So let's say we had a cross that was big A, and we crossed it with a person that was albino. Right? So that person would be albino. Neither one of these crosses would produce offspring that were albino.
But the bottom one could produce a carrier.
Yes, the bottom one would have to produce a carrier.
Right.
So they would be carriers, but they would not be albino. Okay. Does that make sense?
Yep.
Okay. Okay. So let's look at sex determination. So in our sex in a pattern of inheritance in mammals, we say that big X is what is said to be the homogametic sex. So what that means is that they produce only gametes with the X chromosome. So this is the homogametic sex, and this would be female. Okay. So those are our sex chromosomes, our X and Y. And so XY would be the heterogametic sex. Right? So the heterogametic sex would be male, right? So if we look at the way that the X and the Y chromosomes separate during meiosis, we're going to cross a female, and I'm going to put this for my cross because I don't want to confuse it with another X, right? So a male and a female come together, right? So the female can only produce X chromosomes in her eggs, where the male can produce an X and a Y. And so I could put them together in my Punnett square, right? And so on average, you would predict that half of the offspring should be male and half of the offspring should be female. So even though these are X and Y chromosomes, we can still show how they are passed down using a Punnett square, okay? So what I'm going to do is I'm going to show you this little video that talks about what is referred to as the sex ratio. And the sex ratio has to do with like how many males to how many females. And I mentioned before that generally more baby and boy embryos are produced, but over time we see that kind of leveling off. And also the proportion of male babies that are produced also depends upon what the mother is experiencing. So if the mother is under stress, she's more likely to produce a female, and if she's not under stress, then she's more likely to produce a male offspring, okay? So this is a real fast video, and so just keep up the best you can. It's called Minute Earth, and so 2.5 minutes, 2 minutes and 49 seconds explaining it. Oops. It would help if I
Parents-to-be assume that the sex of their child comes down to a flip of the chromosomal form with an equal chance of having a boy or a girl. But in reality, the odds aren't even. For every 100 girls born, the world gains about 106 baby boys. Currently, that skewed sex ratio comes out to roughly 10 million more baby boys than girls born worldwide each year. There are some countries where human meddling stacks the odds even higher in favor of boys, but that doesn't explain the fixed odds everywhere else. The intrinsic boy-girl ratio is rigged by Mother Nature. In fact, it's even more rigged than birth rates show. Human conception results in about 150 male zygotes for every 100 females. But there's a rather tragic reason for this big biological boy bias early on. Male fetuses are much more likely to be miscarried or stillborn than female fetuses. And boys that do make it out of the womb suffer more fatal diseases, take more mortal risks, and fall prey to more violence than girls. So by the time kids grow up and reach baby-making age, the ratio of males to females is just about 1-to-1. But the likelihood of a boy even making it to birth is also influenced by his mom's living conditions during pregnancy. For example, when a massive famine struck China in the 1960s, the relative likelihood of having a son suddenly dropped until the famine ended. And male Americans born to billionaires seem to have higher than average odds of fathering sons. Somehow, female biology suppresses boy survival in the womb during tough times and boosts it when times are good. We've seen the same pattern in other mammals too. When resources are scarce, mothers give birth to fewer males than normal. When resources are plentiful, they bear more. The best explanation we have for this has to do with sex, the other kind. In biological terms, the whole goal of copulation is to reproduce, to pass down your genes to someone who will someday pass them on again. Female offspring are almost guaranteed to reproduce, famine or no famine, because male mammals are pretty much always willing to mate. Males, on the other hand, have to compete for mating privileges. A well-nurtured hunk has a good chance of mating with lots of females, while a male weakened by famine might not score at all. So male offspring are a bigger risk in general. At all stages, they're more likely to die. And even if they live, they might not reproduce. But when times are good, boys' potential to father lots and lots of babies make them a biological risk worth taking. Thanks to everyone who supports us on Subbable. Without you, we literally couldn't continue to make these videos. If you aren't yet a supporter of Minute Earth and want to become one, head over to Subbable.
Okay. So more baby and boy embryos are produced, right? But they're more likely to die. And the reason why they're more likely to die is that if you look at their chromosomes, if they have a bad gene on the X chromosome, they do not have another X chromosome that might be able to balance that. So what that means is that if you have a recessive deleterious gene on the X chromosome, it's going to be more often expressed in boys than in females, okay? So let's look at the example of hemophilia. Oh, here's a picture. So this is an example. This shows the difference in the size. That's the X chromosome. Those are replicated chromosomes that have already copied their material. And then the Y chromosome. Okay. So let's look at sex-linked traits. So specifically, let's talk about an X-linked recessive. And this is hemophilia, specifically A. And remember when we were talking about blood disorders, hemophilia is due to a mutation in a gene that codes for one of those 13 clotting factors. And so what that means is the blood does not clot properly, and so people will start, if they get injured, will bleed. And it used to be that hemophilia was a very bad disease that you would die really early of before even passing your genes on to the next generation, right? But we now have the ability to give people clotting factors kind of to help them survive hemophilia. So if we look at an example of a pattern of inheritance, right, we might see that it is more likely, and this is just obviously too small to see, but I want to show you that it's actually more likely to exist in male than in female offspring, okay? So in this particular instance, I have to use a key that takes into consideration the sex of the individual, okay? So if it is XH, this is normal, right? If you have an X with a little h, this is the allele that causes the disease. Okay. So in this particular instance, the males, you have to make sure that you just draw in the Ys. And so when I'm doing this, I just draw in the Ys right away, because then I don't have to worry about then males and females, right? Because now it depends upon what combination of alleles they have, okay? So what allele must this individual that has hemophilia, which one of these alleles must they have? Little h, right? So this is my genotype, and that means they have hemophilia, okay? So which X do they get? So do they get their X from their father or from their mother? Their mom, right? Because they inherit their Y from their father. So I know that this female has to be a carrier because she's normal, right? But she passed an X with a little h on to her son. Okay. Now the father is normal, so which of the alleles must he have? Big H. Okay. So the question is, could they ever have a female child in this particular family that has hemophilia? In order to have hemophilia, they'd have to have 2 little h's. Is that possible? No, okay? Because this female would always inherit at least one big X, or one big, excuse me, one big H, okay, from her father. And so the question is, what is the probability that she is a carrier? And so you can do a Punnett square that shows this, okay? So for example, I could have this is the father, right? And then this is the mother, oops. Right, and then I do my Punnett square, and it doesn't matter if the mother or the father is on the top or the bottom, but I'll put the mother along the side. I'll put the father up here. Right, and then I combine them. So this is fertilization. These are the offspring. And notice how I always put the X's, the big H first, just makes it easier. Okay. Okay. So now when we're trying to determine probabilities, we need to take into consideration if that individual is female or male. So she's a female, right? So we just kind of ignore these males. So what's the probability that she's a carrier? Not 25%, 50%, right? Oh, we ignore the males, okay? What's the probability if they have a male offspring that male offspring like fed another one, and if they're like, oh, it's a male, because they can see it on ultrasound, but they don't want to do the genetic testing. What is the probability that they would have hemophilia? 50%, right? And then you can say, well, if they had another offspring, and it didn't matter, we weren't concerned if it was male or female, we would say that there's only a 25% chance that if they have another offspring, male or female, that offspring would have hemophilia. Okay. 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 I'm going to skip to this one. Okay. Okay, so the last thing I want to talk about is the inheritance where we have more than 2 alleles. So we have multiple alleles. And a good example of this is the ABO blood type. Okay. Now, we talked about this. This determines whether or not these genes determine whether or not there is A antigens, B antigens, or no antigens in the case of O blood type. And it is determined by a gene, and they've given the gene the letter I. So the gene is I. Okay. The alleles, instead of 2, there are actually 3. So there's I with a big A, I with a big B, and little I. So this is why it's called multiple alleles. So even though there are 3 alleles, we only have 2 in our genotype because we're diploid. So we could look at the combination. So we could look at the genotype. Okay, and then we can look at the phenotype. So I'm going to make a little table here. Okay, so the genotype would look like this. That could be one possible combination. This could be one possible combination. Okay. And I grouped them together for a reason, because if we look at these 2 genotypes, they give us the exact same phenotype, right? So the phenotype for both of those would be type A. Right? So if you did your blood typing and you found out it was type A, you would actually not know whether you had 2 copies of the dominant allele. And notice how there's a little I, and that's recessive. Okay, so the little I is recessive, the big A is dominant. Okay. Same thing with type B. right? There's two possibilities. That's the phenotype. That's the outer expression of the combination of alleles. This one gives us type AB. And so it's important to note that these are both dominant, and so they're both equally expressed. So you would have A antigens on your red blood cells as well as B antigens on your blood cells, right? And then if you are type O, you know that you are this combination. So type O is the recessive combination, right? And so you would know that you would have to have two little copies of the little I gene, okay? Now, the problem comes in is that in our blood types, remember, we don't just have A, B, and O, or A, B, O, right? We have positive and negative. And so this is actually a separate gene. Okay, and so let's go to whiteboard. Okay, so we have what is called the Rh gene, right? The Rh gene can be written in a couple of ways. It could be written as D, right? Or you can write it as RH, okay? So I'll just show you how to do both of them, okay? So we can say that big D causes the Rh antigen to be present. So that would be the same thing as Rh positive, right? So big D is dominant. It causes the RH antigen is present, okay? Little d is recessive. And so this means that the Rh antigen is not present. Oops, present. So you could write that as Rh negative. Okay. So I could write O positive, right? So I could go, O is I, positive is a totally different gene, right? So I could write it like this. So this would be O positive. Right? I could also write this as so that's also O positive, okay? And I think in your homework, I actually used the positive and the negative, okay, rather than D. Okay. So if we look at somebody who is AB negative, so that combination, and let's use RH, RH negative, RH negative. Okay. AB negative. Now, when we look at the way that these alleles separate, in most cases we assume that the alleles are on different chromosomes and they're going to segregate independently of one another. So if I look at my chromosomes, this could be one set of chromosome, and this is my IA, right? And this is my other chromosome, and this is my IB. And then I have another chromosome, totally different shape, right? This is RH negative, and another chromosome, which is RH negative, okay? So that's how the genes appear on the chromosomes. They are on separate chromosomes. They're not on the same chromosome, because that would actually determine how they are inherited, okay? So when we look at this, you can look at how those chromosomes would behave during meiosis. And so we can figure out the gametes that are produced, okay? So question is, what kind of gametes are produced? So I could have this combination, or I could have this combination, right? Because remember that in gametes, you don't have chromosome pairs. So I could have one of those and one of those, and since these are the same, I'm not going to pay too much attention. Or I could have this one and this one, right? So when I'm doing these problems, the first step is to figure out what are the possible gametes that can be produced. Okay. So what about this one? What kind of gametes can this one produce? What would be the combination of alleles? Only one kind, right? So I'm just going to circle these. Right? So what I could then do is I could create a Punnett square, right? But instead of, only one gene, I have two genes. So I'm looking at the inheritance of two separate genes. And so my Punnett square could look like this. Oops, I would only need one. Sorry. Okay, so on top I would put IRH positive. That's a gamete because it only has one copy of each gene. And then this one would have one copy of this gene. And then this one would have one copy of this gene. And then, this is really important, I want to keep the I's and the RH's together, just so it's easier for me to read, okay? So this is my genotype of my offspring. Oops, RH negative. And then I put positive first because that's dominant, okay? So what is the genotype of this individual? Phenotype, sorry. What is their blood type? A positive, right? And so are they like either one of their parents? No, right? Because one parent was O positive and the other was AB negative. And so this offspring is completely different blood type than either one of their parents, right? And that can be explained by looking at how the alleles separate during meiosis, okay? So let's look at this one. I always put the positive ones, right, first. And so what type of blood type does this one have? B positive, right? Again, the two offspring that are produced are totally different from either one of the parents, right? And our expectation was that on average, 50% would be A positive and 50% would be B positive, whereas the parents were totally different, okay? Okay, so that was in your homework as well. Okay, so I think I've covered everything for your homework. So just make sure that you bring that to class on Wednesday to get credit for your quiz. And then we'll continue talking about genetics on Wednesday as well.